Oh, great, we're live. Hello, everybody, and welcome to the Kelp Ecosystem Ecology Network data presentation. I am Jarrett Burns here at UMass Boston. I'm the network coordinator. And I'm going to be presenting to you guys today a little bit about the data that is available from Keen. In particular, we're going to talk about two different data sources. This is a Hangout on air, so we're live. There's a chat window that is open, so anyone who is tuning in virtually, you can ask questions as you will. There are also a few people, I'm looking up at a couple people in the room here who are attending locally here at UMass Boston, and so they may have questions as well. So we're going to kick this right off. Like I said, feel free to stop anytime with questions. I have a chat window open on the side, so I should see it, or someone in the room will flag me. And as this is a Hangout on air, it's going to live on YouTube. So those of you that are interested in the data and want to work with it in the future, you can take a look at what we have here and use this as a set of instructions for how to get access to some really great data on both the kelp forests of New England as well as the kelp forests of the world. So let's begin by screen sharing and hope that the technology gods are kind. Sometimes they are. All right, so like I said, welcome to the Kelp Ecosystem Ecology Network discussion of data. So it's Data Day. For those of you that are tuning in as part of your interest in the Keen meeting that we're having here at UMass Boston over the next few days, tomorrow we're going to be talking about our observational and removal protocols, and then we'll be talking about species identifications on Thursday. But today is Data Day, so the introduction to the data that we have available for you. So I'm going to begin today with just a brief overview of the Kelp Ecosystem Ecology Network, of what it is. Those of you that are tuning in tomorrow, I'll talk about this in a bit more depth. And briefly, the Kelp Ecosystem Ecology Network is a group of kelp ecologists around the world who are interested in understanding the impact of global change on the world's kelp forests. This is a group that has many different members and partners. Just shown here are the coordinators for different regions around the world. So I'm Jarrett Burns. I'm not only the network coordinator, but I coordinate Keen 1, or the Gulf of Maine New England Region Network. And as you can see, we have coordinators around the world who, if you are watching this and are interested in participating in Keen activities, contact me and one of these people, and we will get you looped in. For more information about Keen as we go forward, and for links to all of the data that are going to be discussed today, you can come to kelpecosystems.org, and that is the Kelp Ecosystem Ecology Network website. And there we have much more information about what is going on as part of Keen and how you can become involved. So today we're going to be talking about Keen data, and there really are three different data sources for Keen. The first is from Keen Activities looking at synthesis. So we're going to talk about the Kelp Time database that we developed as part of a synthesis working group at the National Center for Ecological Analysis and Synthesis, and was then published in the Krumm-Hansel et al. 2016 PNAS paper. We also have field sampling and manipulations, and today the Keen 1 data that I'm going to be talking about is from some of our observational field sampling that is done by network members here in New England. And last, we are going to be providing a data product looking at remote sensing of kelp forests from citizen science, and that's from a website called Floating Forests, a citizen science website. I won't be talking about that today. That's data that actually my graduate student, Isaac Rosenthal, has cranking away right now on his computer at home. And instead, he's following the chat here. So, first off, we're going to talk about Kelp Time. Kelp Time is a database of change in kelp abundances over time. This is a database where we went into the literature and into people's individual data sets and actually pulled out kelp time series from around the world. I'm going to talk about both the global perspective and also because this particular meeting is part of a Woods Hole Sea Grant funded initiative, we're actually going to really zoom in on the New England data. So the data from the Kelp Time database came from the 2012-2013 Kelp and Climate Change Working Group at the National Center for Ecological Analysis and Synthesis. So I'm going to start by acknowledging all of those scientists here. This was really a big group effort. And if you are really interested in digging into this database, it was recently used for this publication, Global Patterns of Kelp Forest Change Over the Past Half Century, by Kira Krimhansel and colleagues. So this represents really the first attempt at analyzing this data to look at long-term trends. There are many other different things that can be done with this data. We were simply looking at individual trends separated by different ecoregions around the world. Broadly, the Kelp Time database is a database of 8,000 or so, roughly, measurements of kelp abundance taken by scuba divers. These are field measurements of laminarian kelp, so this is specifically laminarian kelp. We do not get into other taxa, but really try to restrict ourselves here. Although for those of you who are interested in potentially other taxa that could be within the database, we do have raw data, and it is possible to extract out some additional information, say for some fucoids in select regions, as well as some Saccharija. Broadly, what the data looks like is this. So here, for example, we have 3 potential sites. There are data points for every year. And given that we have a number of different types of measurements, kelp biomass, percent cover, stipe abundance, individual abundance, we've created standardized metrics of all of these. So broadly, we can actually compare changes in kelp abundance using a standard metric across all of these very different data sets. And so here, for example, is an analysis, a sort of theoretical analysis, where you have a linear fit to each of these time series, and the dashed line is a fit from, say, a Bayesian hierarchical model like we used in the Krimhansel et al. Paper. Okay. So, some information about Kelp Time. We have a lot of data, over 8,000 measurements, as I mentioned, and that comes from 1,400 sites. Here, what I have is just a map drawn directly from the database itself. And within this map, you can see that the warmer colors represent more sites per region. So again, we're using the Spalding et al. Ecoregions. Cooler colors represent fewer time series per site. And we had a cutoff of a minimum of 3 years in order to consider a single site valid for this database. So looking at this map, you can see a lot of things. For those of you that are interested in getting out there and collecting new and original data on kelp forests, you can immediately see that, hey, there are some regions of the world that are very undersampled. In fact, gray regions are places for which there are kelps, but we have no data. So get out there and sample. You can also see that the data coverage is highly uneven. So there are some places that we have a ton of data. Southern California, Northern Ireland, Tasmania, and other places where we could do a little bit better. But uneven coverage, but it's there. There's a really good spatial coverage around the world for this data set. Most of the trajectories within this data set are roughly 10 to 20 years. So we have study duration along the x-axis and the number of sites on the y. So this is a histogram of study durations. You can see a nice peak there around 15 years. But you can also see that we have data sets within this particular database that are up to 62 years long. So we do have some fairly long trajectories. That's not necessarily 62 continuous years. There are definitely gaps within some of those data sets. But we do have a number of longer data sets as well as shorter ones. Here's just sort of a histogram again of when samples took place. And you can see, again, most of the data sets that we have come from more recent samples, so the 90s and early 2000s. There is a drop-off as we get into 2012, and that's primarily because we stopped collecting data then. So if something had not been published yet, even if sampling was ongoing in 2012, it's not there. I will say, and I'll mention this again, for those of you that do have Kelp Time series and are interested in contributing to the Kelp Time database, while we have a current published version for Krimhansel et al., we are interested in updating this database. And so over time, we'd like to see this database grow. If you either have a data set that's in here and would like to add additional data, or if you have a time series that you feel like we missed or just didn't see and you would like to add it to the Kelp Time database as well, please contact me. We would love to add your information to this data. That can be anything from your own personal data to agency data, et cetera. We're really trying to cast a wide net here. So here's what the whole data set looks like. We have year on the x-axis and standardized kelp abundance on the y. And this really gives you a sense of when we had samples taken. It gives you a sense of the variability. That shaded region is the confidence interval, or rather the credible interval from a hierarchical model of best fit. But there's just a huge amount of variability here. We found in the Krum-Hansel et al. Paper that the most useful way to look at this data set was to actually look by individual ecoregions. And so you too can do this. If you're interested in accessing this data, and that's after all what this presentation is about, you can get access to this data at the Temperate Reef Base. So Temperate Reef Base is a wonderful data site that has been set up by our colleagues Craig Johnson and others down at the University of Tasmania as part of the Integrated Marine Observing System. So it's not just our data that's there, but quite a few others. And so what I'd like to show you guys now is actually how you can access the Kelp Time database, so you too can go home, download some Kelp Time series, and analyze them yourselves. So with that, I will set screen sharing. Stop that. And I'll move the screen sharing over to a website. So this is the central portal for Temperate Reef Base. Hold on. So this is what you get when you go to temperatereefbase.imas.utas.edu.au. And if you want to access data from the Kelp Ecosystem Ecology Network, first click through to get data. Also, before I move on, I'll notice there is this submit data button here as well. If you've got data, and it doesn't just have to be about kelp forests, but any temperate reef data, from observational data sets to experiments, we want your data at Temperate Reef Base, so please contribute. So we click through Get Data. And you'll notice immediately, actually, we see a lot of other data sets. The Reef Life Survey is currently hosting their data on Temperate Reef Days, so you can access that there. And there are, excuse me, a wide variety of other data sets available to you. To get access to the Keen data quickly, we can just search Keen as a keyword. And here we have both a global Kelp Time series from the Kelp Working Group, as well as some work, a data set on fish abundance that Alejandro Perez Metu and I are currently working on as a follow-up to that working group. So we can click through and select that data. And what I really like about this website is that you end up getting a interface where you can choose the data that you want. You can just download it all by immediately going down and clicking Next. Or you can select a subset. So for example, because today we're really focusing on New England, let's choose this subset right here. Great, we've got a box highlighted. I could highlight other boxes if I wanted. And then Next. So here we have the download page. There are a couple of options present here. We have a means to view the metadata record. So this is all ISO compliant metadata, so you can get a full metadata set to work with, as well as to understand exactly what each of the columns within the resulting data set will mean. Or you can choose how you want to download the data. If you want to get access to the full data set, these two links are to the aggregated and raw clean data, but we're just going to download a CSV file right now. You'll notice that this data set is currently under CC BY, so we would ask that you cite the Krum-Hansel et al. Paper if you're going to use the current version of Kelp Time going forward. So I understand. Download it. And then we can take a look at it. All right. Let's stop that screen sharing and switch over so we can actually take a look at the data. And you'll see broadly, there's a lot of information here. Let's see if we can increase the zoom. So everything from study identifiers, study names, sites, geospatial information, depth, etc. But rather than go through the raw data and get lost in all of these numbers, I'll kind of talk about this a little bit more. So I'll go back to our presentation. There we go. Okay. Broadly, the data that's available from Temperate Reef Base is a number of different metrics of summed kelp abundances. So I say summed because we went through and summed over the kelp abundances of all species in a given year. So these really represent site level values. We have biomass, stipe abundance, individual abundance, and percent cover. And different data sets have different types of values. Each time series has a unique ID, so that's fairly easy to sort on. And there's geospatially explicit information for all samples. And we also, as you saw, have depth. So here I've actually plotted out all of the sites that you find from that New England data set that I downloaded. So this is something I just whipped up in R with ggmap. And for those of you following along at home that are interested, I used the Wes Anderson color palette package to use the ZZoom package palette, because, you know, it's ZZoom. Hopefully share my humor in using that. Maybe not. Anyway, so here we've got all of the sites that we have from New England. You can see we've got a pretty good spread. From the Krumhansel et al. Paper, we saw that within the Gulf of Maine proper, we had a fairly high degree of certainty that there were declines in New England, although if you got further north, it actually looked like there were increases. So there really was a lot of variability across this entire analysis. And you can see that if we plot this. So here again, those colors should be the same as before. You can see that some of the longer data sets, particularly from New England, show some declines. So you've got some these longer data sets dating back into the 60s. You can kind of see the decline of kelp as urchins started to become dominant in this area. You can really see these in these red colors. These are some of John Whitman's sites, for example, that really tanked due to the influx of urchins. You can see in the late 90s and in the early to mid 2000s, there was a much higher degree of variability. Although for the Gulf of Maine, our model said that on average, it seemed like that while there was probably some recovery at some sites, it was sporadic, and the overall regional trend was a mild decrease, or at least a fairly well-supported decrease in kelp. Anyway, that's the kind of analysis that you can do from the kelp time data set. You can do it for your region. You can do it for other regions. There are a lot of possibilities, and particularly if you can bring other environmental covariates to bear, there's a lot of stuff you can do with this data. Now, I did mention that we summed across a lot of different species within that data set. If you are interested in getting access to not only the original data that we used for the kelp time data set, but also even to take a look at some of our analysis, the entire framework is available on GitHub. So if you were to go to github.com/kelpecosystems/global_kelp_time_series, it's a mouthful, you actually have all of the raw data and code from this analysis. There is a zip file in here, and that zip file is all of the raw data. And when I say raw, I don't mean like raw, as in what our data contributors gave to us. This is a clean data set that we've actually cleaned up so that it is standardized. So every file is the same and has the same structure, so you can merge and put together different data sets. What I do mean by raw, though, is that this data set actually has individual plot level information. And this data set also has individual species level information. So we haven't aggregated across species. So we have data at the quadrat or transect level. There's often replication within years. We have species level information for most of the data. Sometimes it is still just kelp. And because of this, you can create much richer analyses. So here, for example, I've plotted John Whitman's data again. And John was actually looking at a couple of species of kelp. I've highlighted the two for which he has the best records. We have number per square meter, so you can see we're back in natural units again. So we're no longer at standardized kelp. And we have Alaria esculenta, the feather kelp, which you can see was in pretty low abundances, but there was a sort of a blip up and then it dropped down again. And then Saccharina latissima, the dominant sugar kelp here in the southern Gulf of Maine. And you can really see the decline in Saccharina. You can see that some sites that were sampled in the middle did show some blips around, but overall the trend really has been long-term decreases from John's data. And that's from the early 1980s to the present. Okay, so that is the kelp time database. And I'll just take a quick pause, get a drink of water. All right, so I'll talk next and finally about the field sampling data, and particularly the field sampling data that we have from Keen 1. I'll talk about what's there and how to get access to it, as well as how you can start to use it. And there are a lot of different ways that we can use this data set. It's a pretty, even with the sort of several sites we have already, it's already getting to be a large data set, and I'm hoping in the coming weeks we can begin to bring in some of our non-New England collaborators and expand this out further. And again, I won't be talking about our remote sensing data today. That will be for another time. So first off, what is the Keen observational sampling? Well, in the Keen observational sampling, scientists and their dive teams head out to reefs. These are reefs that are at depths between about 8 and 12 meters, although we've actually gone a bit shallower for some reason, for some reefs in some regions. We look for areas where kelp generally is located, and then implement a stratified random design where we split sites into 4 areas and then randomly sample within those areas every year. So, and then we resample annually, where we take new points every year. So those points do move around. These are not permanent transects, but we do sample the same sites year after year after year. So we get a sense of what is the average abundance of a variety of different taxa at each site in each year. We use a combination of different methods along 40-meter transects, and I'll talk about those in just a moment. But broadly, I'd like to acknowledge the Santa Barbara Coastal LTER, the New Zealand MPA sampling, the Reef Project, and the Tasmania MPA monitoring and PISCO, all is providing input and techniques that we have adapted into the sampling scheme. And so this is all diver sampled. So this is all diver sampled data. These are biodiversity samples that are taken on scuba. There's also temperature data that goes along with these, although those are not yet available. We're still working on cleaning those. There's a little bit of reprocessing that still needs to happen. So Keen 1, the Kelp Ecosystem Ecology Network of New England, has launched its first set of data. I'll show you where it is so you can get access to it in just a moment. Broadly, the data that we have listed right now comes from 4 labs: my lab at UMB, John Grabowski's lab at Northeastern University, Jen Dijkstra's lab at UNH, and Austin Humphrey's data, lab at URI. We're also going to be adding data from the Rasher lab and the Price lab. They're both up at Bigelow. So we should have some more data down east further this year. If you are a marine ecologist in New England and you want to jump in on this, please contact me. We have these meetings every year to make sure we're still on the same page. And every year some small tweaks happen. So we would love to add more sites along the coast. But you can see currently we span a pretty big span, all the way from the mouth of the Narragansett in Rhode Island up into Maine. And as I said, we're hoping to get further down east later this year. So it's a pretty wide geographic span that also spans a fairly large temperature gradient. The raw and processed data is currently available on GitHub, again at github.com/kelp-ecosystems, and then /observational data. And I'm just gonna, I'm gonna walk you through that really briefly. So let's stop that screen share and kick in the next one. So go back to this window with temperate reef base. And so this is the Kelp Ecosystems GitHub repository. We've got a couple of different things here. This materials repository is actually where we have the handbook for sampling and all of our materials and methods if you want to dig into it. In observational data, you can see that we have a folder with the raw data. So that is the data as given to me by PIs with some light cleaning and scrubbing of typos that I've noticed along the way. And also I've provided the full data cleaning pipeline in this R folder. Ultimately, the data that you're going to want is in this clean data folder. And you can see we've got a couple of different types of data. We have information on sites. We have information on swath sampling, quad sampling, fish sampling, and cover. And so for any one of these data sets, you can browse it just by clicking on it and taking a look. And then you can download them as you will. So here, for example, is our site data set, and you can see our format as well as all of the columns. We'll be providing some more metadata documentation for this in the near future. Okay, so back to the presentation. One thing we provide is a species list. So every region has a standardized species list with species codes in them that are used on the data sheets. These species lists basically say what is sampled and what protocols are used to sample a given species. The taxonomy is all standardized. So while the taxonomy within the species list itself is a little fuzzy, before merging it with the data sets itself, we query the a global database to standardize taxonomy, the WORMS database. So you'll see within these data sets a nice standardized set of taxonomy. The data, as I showed you before, is in a long data format, and that means that every observation of every species has a single line. So those of you that are familiar with the wide and long data paradigms, it's long data. It can be reshaped very easily into wide data, but you can use this data in a number of different ways. So pull data from the clean data. And I'm just going to walk through really briefly what each data set provides. So the site data is the key that you can join on all other data sets. It has the latitude and longitude of each transect in every year, as well as accompanying day of physical data, so typically water temperatures and visibility, as noted by the divers in the water on the day of sampling. Again, that your mileage may vary in terms of local conditions. We have some sites that vary a great deal from day to day here in the Gulf of Maine. Fish sampling. So the fish sampling technique broadly are swimming up transects, looking at 1 meter on either side of a transect tape, and 2 meters up. We count all fish off the benthos and record them in size bins as well as young of the year. So here is an example of the kind of data you can see in the Keen 1 data set. Here I have counts of cunner that are 0 to 10 centimeters and 10 to 50 centimeters. These are number per transect. And I have the sites arranged roughly from south to north along the x-axis. This is pooled across all years, but you can begin to see at least some broad trends. For example, we tend to have a lot of smaller cunner in Fort Wetherell, or at least in the 1 year that was sampled relative to other sites. Second, we have the Quadrat data set. Within the Quadrat data set, every transect has 6 quadrats, each is 1 square meter. These are largely for solitary large algae, invertebrates, sometimes cryptic fish are included, some mobile species as well. Basically anything that you can count as an individual and that is common enough that we can pick it up in a quadrat. So here, for example, is another way you can look at this data set. So here, what I'm looking at is the time series of urchin abundances across all sites. Color corresponds to site ID, and I've made symbol to show you PIs. We've got number per square meter on the y-axis. And you can see that when we started doing the sampling, we really did not see a lot of urchins across sites in the Gulf of Maine. It's really only in the past year or so that urchin abundances seem to have gone up. It's only 3 years of data, so who knows what that's all about, but it's fairly striking. So it's really easy to get time series like this. Each dot is a mean, and the error bar around it is a standard error. So it's, again, one of the nice things about working with this long data is that it's very easy to summarize and make these kinds of plots. Swath sampling is similar to quads. We do 4 1 by 20 meter swaths along the transect line. We'll talk more about this tomorrow. And this is largely for slightly more rare species. So here, for example, is another way. So this, I mentioned before, we have long data. I've actually reshaped the swath data into wide data. And for anyone who's interested, I'll provide the script that I use to do this. And here you can see a plot of the relationship between rock crabs, Cancer irroratus, and Jonah crabs, Cancer borealis. And you can kind of see the inverse relationship between the two of those species across all of our sites over all time. So again, really easy to look at by plots like this. And because sites have common site names and years, you can join different data sets together to generate these kinds of comparative correlative analyses. Last, we have uniform point count. So along every transect line, we sample 80 points. And under each point, we note the presence of all species under the point. So what species are present under 80 points, as well as the substrate type. From that, we're able to derive an estimate of percent cover of different species. You can also use a data set like this to begin to look at something like sessile species richness. You can look at algal species richness. You can look at sessile invertebrate species richness. So here, for example, I've aggregated all of the species richness numbers. So this is total sessile species richness on the y-axis. And on the x-axis, we have the percent cover of sugar kelp, again, as derived from this point count technique. And you can see, again, there are different dots or different colors, rather, or different sites. And this is kind of a cloud, but it suggests, if you start to squint and look at this, there are probably a lot of different patterns here that you could begin to look at. Not only that, but because we're looking at sessile species cover here, there are a variety of different ways that we can look at this data. And some of the species that are recorded are also recorded in quads and swaths. So we can do things like here I've merged together, or here I've plotted the side-by-side results looking at percent cover on the left-hand side of this plot, and the number per square meter from quadrat sampling on the right side. And you can see that we get slightly different results. So we need to sort of think carefully about how are we looking at kelp abundance using this data set. So a lot of interesting information here for comparative analyses, and also for teasing out why one of these may change in one direction while the other may change in the opposite direction. I wanted to end up to talk a little bit about data sharing, and then I will stay online. I'm going to end this Hangout, but I'll stay here with the chat window open in case any of you have additional questions, because the chat will be archived permanently along with this presentation. And just talk for a minute about data sharing. So first off, data is open within the network. And as a data contributors commit to their data being fully open after the first publication. So within the Kelp Ecosystem Ecology Network, once we publish your data, or somebody within the network, presumably you, publishes that data, then it's open. We will be providing it on a temperate reef base. So you can see that with the Kelp Time database, for example. It's now there, it's available. Observational data, as we created the network, everybody felt very strongly that data is immediately useful, for example, to management agencies. So that observational data, once it is cleaned and merged in with the rest of the data, is open outside of the network. However, because we are using a Creative Commons BY, we actually do request that collaboration on data that has not yet been seen in academic publications be requested. So if you're going to use my data, for example, of Appledore Island in 2016, and I haven't published it yet, we ask that you request collaboration at any point. So please do that. After data is published, of course, we ask that you cite the individual data sets, and in working with Temperate Reef Base, we're going to be providing citations, so citation information, so that you can cite individual scholars for their data contributions. We really want to make this data useful for everybody's professional CV. And as I mentioned, data will be available with ISO compliant metadata and citation information on Temperate Reef Base shortly. In putting all of this together, we've gone through the final round of data cleaning for the Keene 1 data. And so it's beautiful and shiny. And so we'll be putting that up shortly. And then as other collaborators contribute their observational data, we'll be making that available along with citation information as well. That's it. And so I'll take some time for questions. If there are any questions from the room, or if there are any questions online, although my online may have stopped refreshing. No. Otherwise, you're welcome to contact me at jarrett.burns@umb.edu. That's JARRETT.burns, BYRNES, @umb.edu. I'll hang around for a few minutes on chat after I end this Hangout, and if you have questions about working with the data, or if you want to download and begin working with the data yourself, please let me know. All right. And I'll end that in particular by thanking all of the different contributors to the Keene 1 dataset over the years. This is a picture from one of our meetings a few years ago. Thank you very much, and I'm looking forward to many of you being back and attending tomorrow's meeting for our annual protocol coordination. All right. Thanks so much. I hope you enjoyed this, and I hope this is useful to you, that you find this data meaningful, useful, and it helps out your research. And I'll hang around a little bit on chat so we can see what questions people have, and we will go from there. All right, thank you very much.